Welcome back, students. Now in this section I'll be talking to you about the biosynthesis of non-essential amino acids and also the biological importance of the different amino acids. We saw how they are catabolized. Now let us see what is their how they are synthesized, how the non-essential amino acids are synthesized. So if you look at it, most of the non-essential ones can be synthesized by transamination. That is a central reaction. So which are the ones which can be synthesized by transamination? These include alanine. Again, from MCQ point of view, I am talking about this. Alanine, aspartate, glutamate, all this should be known to you. Synthesis by amidation, that is adding an amino group, glutamine and aspargine, glutamate to glutamine, aspartate to aspargine. From intermediates of the TCA cycle, we have proline, serine and glycine that are taking place. And from precursor essential amino acid, cysteine and tyrosine. Again, I have told to you, cysteine is got from methionine and tyrosine is got from phenylalanine. Two central enzymes are required in this formation. Phenylalanine to tyrosine requires phenylalanine hydroxylase. Methionine to cysteine requires the formation of homocysteine and then cystathionine beta synthase, further cleavage to form cysteine. And plus it requires the amino acid serine, only then methionine can form cysteine. So with this we'll proceed, and also remember that when we say precursor essential amino acids, cysteine and tyrosine, sometimes these will become essential if the precursor, there is a deficiency in that enzyme. Suppose phenylalanine hydroxylase is not there. Phenylalanine is also essential, at the same time tyrosine also becomes essential. If cystathionine beta synthase is deficient, methionine also becomes essential, cysteine also becomes essential. So now we finished how they are synthesized. Just from an MCQ point of view, I have given this. Other than that, one important is synthesis of serine also takes place from 3-phosphoglycerate. So it forms 3-phosphoserine to form serine. So let us go with the biological importance of the different amino acid. So glycine, where is it used for? What is it used for? How is it useful within the body? Is it useful or not? I think by now you should be able to tell that certain proteins contain glycine. So give an example. Collagen. Yes, collagen is a structural protein present in our body. Glycine XY. So glycine is an important amino acid which is present and which is responsible for the structure of the collagen the way it is. So glycine is present in one important thing is collagen. Other than the protein, so all the amino acids are required for protein formation. All the 20 of them in one way or the other are related to protein. So what are the other importance of glycine? Glycine is used for the synthesis of 3 important compounds, and they include heme, creatine and glutathione. GSH, glutathione. These HCG. These are the 3 important things. Other than that, I'll be talking to you what is its importance. So glycine is used in the synthesis of creatine, heme and glutathione. What else is it used for? It is used for the synthesis of purine nucleotides, because in purine nucleotides C4, C5 and N7 are come from glycine, glutathione. It is also used as a conjugating agent. Where have you heard of it? You should have heard by now that bile acids are conjugated with glycine or taurine to form the respective bile salts. Give an example. Cholic acid is converted to glycocholic acid. So where did the glyco come from? From glycine. Quinodeoxycholic acid is converted to glycoquinodeoxycholic acid. So that also comes from glycine. So it is used as a conjugating agent for the synthesis of the secondary bile acids, bile salts, or the primary ones, primary bile salts or the secondary bile salts. It is also used in an important reaction, that is conjugating of benzoic acid. Now why is it important? Benzoic acid can form hippuric acid, and it is an added preservative which is added for many food additives. Hence benzoic acid detoxification assumes importance, and hence glycine is used as a conjugating agent. It is also by itself a neurotransmitter, especially in the brain stem and the spinal cord. It is present in proteins like in collagen. Every third amino acid is glycine. So I'll talk a bit more about the synthesis of heme. I'll be talking completely differently because they are all the porphyrias and the heme synthesis come together. Now to talk about creatine. What is creatine? You must have heard about creatine a lot. You might have heard about creatinine also. So what's the difference between creatine and creatinine, and what are the amino acids which are required for the formation of creatine? Now creatine is creatine kinase also you will have heard of already in different enzymes. So let us look at the synthesis of creatine. Now, creatine synthesis requires 3 amino acids. They are glycine, arginine and methionine. So, what happens in this? Creatine is mainly present in the muscle. It is the main storehouse of energy in the muscle. How can it store energy? Creatine, I'll just talk about creatine. Creatine is converted to creatine phosphate. Whenever it wants, it forms creatine phosphate. So when it forms, from where does it do? Whenever there is extra ATP, all this ATP will give more and more of its phosphate group and creatine phosphate is formed, and ATP is converted to ADP, so that it won't inhibit the other pathways which help in the formation of ATP. So ATP production will continue and creatine will act as a storehouse. So when it release this, when will this reverse take place? This reverse of this creatine phosphate to creatine takes place whenever the muscle requires a burst of energy. Like for example, following a strenuous exercise. So during that exercise, there is lack of oxygen. So maybe the electron transport chain will not be functioning properly and the ATP formation may be decreasing. So at that time, creatine phosphate acts as an immediate source. Immediately it can form whatever ATP is required for the muscle. So it gives that extra burst of energy that is needed is given by creatine phosphate. So where is creatine phosphate and creatine present? They are present in the muscle. And this enzyme is a creatine kinase. Have you heard of this enzyme? You should have been hearing by now because there are 3 isoenzymes of this. CKMB, CKBB and CKML. Now right now what I was talking of? The muscle. So it is CKMM in the skeletal muscle. CKMB is present in the heart tissue and CKPB is present in the brain. So, but in the brain, what is its function is not so clearly known. In the muscle, it is very clearly known that creatine to creatine phosphate, creatine phosphate to creatine. And for this synthesis of creatine, 3 amino acids are required. That is glycine plus arginine. Now what will happen is glycine plus arginine will form what is called as guanidinoacetic acid, and this undergoes methylation by S-adenosyl methionine to form creatine, and creatine to creatine phosphate. What else happens to creatine phosphate? Creatine phosphate can further undergo a non-enzymatic anhydrous removal of water molecule from creatine phosphate, and it will result in the formation of creatinine. Creatinine is the anhydrous form of creatine. Anhydrous, water has been lost. It undergoes cyclization and creatinine is formed. This creatinine can be excreted in the urine. Creatine also can be excreted in the urine. So the muscle proteins, as they undergo turnover, as they keep on changing, some amount of creatine and creatinine are released into the bloodstream, from where they go to the kidneys, they are filtered and they are excreted in the urine. So we'll talk about the importance of creatinine and creatine in the diagnosis of various diseases, how they help in the how their levels are affected. So creatine is present in the muscle. As I said, the first step, synthesis of guanidinoacetic acid, takes place in the mitochondria of the pancreas and the kidney. What is the importance here is, this guanidinoacetic acid is taking place in the mitochondria of the pancreas in the kidney. And where is this present? this is present in the muscle. So something has to transport this to this, and plus in the between it has to undergo methylation. So actually guanidoacetic acid goes to the liver. In the liver it gets methylated. Once it gets methylated, it is transported to the muscle, and in the muscle it acts as a storehouse. Okay, the methylation reaction takes place in the liver, and then it reaches the muscles, and in the muscles it is phosphorylated to form creatine phosphate. The enzyme is CK creatine kinase. It acts as a store for ATP. Now this reaction, creatine to creatine phosphate and vice versa, is called as the Lohmann's reaction. Again, from an MCQ point of view, I am emphasizing this. This reaction is known as the Lohmann's reaction. Please remember that this is taking place in the pancreas and the kidney, and this reaction is taking place in the liver. MCQ point of view. Okay, now let us go to creatinine. So creatine phosphate undergoes non-enzymatic spontaneous conversion to creatinine, the anhydrous form. Now the creatinine is excreted in the urine, and the amount that is excreted in the urine is fairly constant. That is, we can use this creatinine concentration in the urine. We can estimate how much is creatinine is excreted, and we can use it to find out what is called as the reliability of a 24-hour urine collection. What do I mean by this? Now, in many diseases, it is essential to collect urine for 24 hours. Once we collect for 24 hours, the other things are estimated in this 24-hour urine. Now, what will happen in certain cases? Maybe it is an infant, maybe that person is mentally retarded or mentally ill. In these people, they may say that they have collected for 24 hours, but it may not be containing 24-hour urine. What if the urine volume is less? You don't know whether it is because of a disease or it is because that person is not collected. So in these cases, we estimate the creatinine levels in the urine. So it tells us it is fairly constant because it depends upon the muscle mass. More is the muscle. Like in males, if it is muscle is more, creatinine excretion is more. In females, muscles is less, creatinine excretion is less. Normal values are there. It falls within the normal value. We come to know that it is a 24-hour urine sample. So to know the reliability, we use it. Plus, there is one more thing in which creatinine is used. Remember, creatinine is neither secreted nor absorbed from the renal tubules. So it can be used for one of the renal function tests called as creatinine clearance test. So we have the physiologist will be telling you about it. The main thing is formula is U into V by P, where U stands for concentration in the urine, V stands for volume, and P stands for concentration in the plasma. If we know this, we can use creatinine to find out the glomerular filtration rate. So here I have got it. So creatinine is usually constant and it is more in males. We have creatinine clearance test, and urinary creatinine is used for the reliability of a 24-hour urine collection. So the normal serum creatine levels are 0.2 to 0.4 milligram per dl, and normal serum creatinine levels are 0.7 to 1.4 milligrams per dl. That is regarding creatine. Now one more important compound I told you was porphyrins, that is heme. Now compounds that readily bind to metal ions are called as porphyrins. Now why do I say heme as a porphyrin? Because heme can readily bind to a metal ion. Which is a metal ion? Ferrous ion. So it can bind to ferrous ion, and hence it is called as a porphyrin. Now glycine is a precursor of heme. I'll be talking more about heme when we talk about porphyrias. Heme is required not just for hemoglobin. It is also required for cytochromes, hemoglobin, tryptophan pyrrolase, catalase, peroxidase. So many enzymes require heme. Heme synthesis requires glycine. So glycine is important for the synthesis of all these compounds. Now porphyrias are diseases due to defective enzyme in the heme biosynthetic pathway, in which again there are two types, erythropoietic and hepatic. I'll be talking about the porphyrins and porphyrias in a separate class, because we need to know about the synthesis of heme. So we finished one amino acid, glycine, and its importance. So let us go ahead with the second amino acid, that is serine. Now, as I said, one more important compound got from glycine is glutathione. What is glutathione? It is gamma glutamyl cysteine glycine. So in that, gamma glutamyl, glutamic acid is required, cysteine is required, and it also requires glycine. I'll be talking more about the glutathione separately because it is involved in free radical metabolism. So creatine we finished. Now let us go on to serine. The next amino acid that we'll be talking about is serine. Now serine is basically a one carbon donor. MCQ point of view, one carbon donor. It is used for the formation of cysteine. We already saw how homocysteine plus serine, cystathionine, which breaks down to form homoserine and cysteine. So it is used for the formation of cysteine. It is used for the synthesis of phospholipids. Phosphatidylserine is one of the phospholipids that is synthesized. It is also used for the synthesis of choline, and choline is required for the formation of acetylcholine. So serine gets converted to ethanolamine. Ethanolamine gets converted to choline. So choline by a lot of methylation reactions, again methionine donates the methyl groups and converts ethanolamine to choline. Where have you heard of choline? Choline is a vitamin-like factor, true, but it can also be synthesized within the body, and choline is used in acetylcholine formation. Now serine is also present in various phosphoproteins, glycoproteins, etc. It is present in the active center of many enzymes. So the enzymes which contain serine at the active site are called as serine proteases. Why do we say proteases? Most of these enzymes which contain serine are used in the hydrolysis of proteins like trypsin. Trypsin is a classic example of a serine protease. There are what are called as serpins also. Remember, serine protease inhibitors. Serpins are different, which inhibit this enzyme is a serpin. But which contains this enzyme, which contains serine. Enzymes which contain serine are serine proteases. That's the difference between them. Serine is also used for the synthesis of selenocysteine. So let me talk a bit more about selenocysteine. Serine is required for the synthesis of selenocysteine. What is this? Selenocysteine. How much have you heard about it? Now selenocysteine is considered as the 21st amino acid. Till now we kept on talking there are 20 amino acids, 20 amino acids, 20 amino. Now I am talking of a 21st amino acid, and that is selenocysteine. What is this? Usually selenium is a metal. Selenium plus ATP will result in the formation of selenium phosphate plus AMP plus Pi. What happens? Now serine will react with this phosphorylated selenium to form selenocysteine plus inorganic phosphate. Now why do we call any amino acid as a standard amino acid? Now why am I saying that selenocysteine is a 21st amino acid? Thing is, anything which is coded by a codon, then you call it as a standard amino acid. Now what has happened? Is there any codon for selenocysteine? If it was there, why it is not there in the genetic codon table? That question will come. So basically this is selenocysteine incorporation into proteins during translation is a type of different modification is happening. One of the stop codons, instead of acting as a stop codon, will start incorporating selenocysteine under specific conditions. So let us just look upon it. I know it looks as a big thing what I am talking. See, selenocysteine is also abbreviated as selenocysteine or SEC or U also. Now this is incorporated into protein during translation. That is why it is the 21st amino acid, because it is incorporated during translation. Now it has a tRNA which is specific for selenocysteine. Okay, it has a tRNA, fine, it is which is specific for selenocysteine. But what about the codon? Now the codon is actually UGA, which is actually a stop codon. But here in this case there occurs what is called as translational recoding. What determines this? Now there is a specific sequence on the mRNA. Suppose there is a specific sequence on the mRNA, what we call as selenocysteine inserting sequence. It is abbreviated as SECS, selenocysteine inserting sequence. Suppose this mRNA is there. Following this, it has this is the mRNA which is having this SECS. That time, if there is a stop codon UGA here, even if there is a stop codon, but if it is having a selenocysteine inserting sequence in the mRNA, it won't stop. This will give it a signal to the tRNA to incorporate selenocysteine into the protein. So these are called as specific sequences known as selenocysteine incorporating sequence. Now, where is selenocysteine present? There are certain proteins in which selenocysteine is present. Many proteins are known nowadays which contain selenium or selenocysteine. Now, few examples are given again from the point of view of MCQ. Thioredoxin reductase, glutathione peroxidase. This is something which you all might have heard. Glutathione peroxidase is involved in free radical metabolism. So hence you should be knowing this name, glutathione peroxidase. Deiodinase, where have we heard of this? This is present. Deiodinase is responsible for the conversion of T4 to T3. So again, selenium is involved in thyroid metabolism. For the formation of active hormone, we require selenium or selenocysteine. So both are required. Selenocysteine is required for the enzyme deiodinase, which is required for the hormone synthesis. There is also one more protein by itself known as selenoprotein P, which also acts as a antioxidant, and thereby selenium is mainly involved in antioxidant metabolism. It is responsible for converting free radicals to their non-reactive portions, to the non-reactive counterparts. Hence, selenocysteine is very important, and this is known as the 21st amino acid. There is one more 22nd amino acid, but let us talk about it later. So this is what is important regarding selenocysteine. Please remember, they have to be having a specific mRNA sequence known as selenocysteine inserting sequence. So next I will be talking about the other amino acids as we go ahead in the next part. Thank you.